Well, hey biology, hope you're doing well when you watch this video. So this is 6.2, and this is all about chemical reactions, okay, and crazy important molecule enzymes, okay? So we're going to talk about how chemical reactions affect chemical bonds and describe how energy, what it is and how it affects how a chemical reaction will occur, and then enzymes and how enzymes work, okay? So living things are made of compounds, but chemistry isn't just what life is made of. Chemistry is also what life does. And so everything that happens in an organism. So think about it, your growth, your interaction with your environment. If you think about it, how you're feeling right now, if you're happy or sad or stressed, that's because of reactions and different molecules reacting and being around. Reproduction, movement, those are all based on chemical reactions. And if you take anatomy, you're going to learn about myosin and actin and how they interact with each other and deal with ATP and stuff, so chemical reactions. So it's all very important. Chemical changes, it's when they're reorganized into different substances. So in reactions, chemical bonds are broken or formed. And so that results in one of two things: production of heat or light, or the formation of a new gas, liquid, or solid. And so examples of chemical reactions like rust, of course, fireworks or anything that releases heat. This is chemical change up north, right? Leaves changing in the north, beautiful scenery. That's all chemical reactions. Physical changes, I don't know if you ever had a test that you did poorly on and you ripped it up. That's not a physical, that's not a chemical change, sorry. That's a physical change. And so that is not a chemical change. So that's just physical changes. It's not rearranging atoms or bonds. And so, of course, we talked about the famous volcano experiment when people do when they're young, and how changes in the chemical bonds that form that do various things. But you have vinegar and sodium bicarbonate, and then you add those two together, and you notice you're rearranging atoms and molecules and creating new ones, right? Water and carbon dioxide, and you're rearranging stuff. So again, on the left-hand side are the reactants, on the right-hand side are the products. And so that's the famous experiment volcano, right? And you get all that gas, and then you have the explosion, and you make mama happy. And so metabolism, in class, I had you write down a few things, but this is the sum of all the chemical reactions. We talked about ice cream and how certain people process fats differently, but that's not what I'm really talking about here. I'm talking about all the chemical reactions. We all have active metabolisms. And so catabolic means breaking down. You need to know these terms, catabolic versus anabolic. So what you do is you eat or ingest, food, and you break those things down to get building blocks. Okay? And then, you get some energy from that, usually ATP. And then anabolic means building. And so you build different macromolecules from the building blocks. So again, how important is food? Food literally makes up your tissues and your body. So do you want to be built with Doritos or, lean protein and broccoli? So it's a difference, right? It's very important what we input into our body. So a chemical reaction, we talked about this already. On the left-hand side, what goes into a chemical reaction are called reactants, and what comes out are called products. And we talked about the example, I believe, like French toast, right? So you need toast, you need eggs, you need sugar, some butter in the pan. Those are all the reactants, and the product is a glorious tasting French toast. Here's a famous example, need to add this as well, but here's a famous example in biology. You have glucose and oxygen gas, and then that yields CO2, which is waste product, water, which can be used, and the star of the show would be ATP, energy. Okay, so we take the energy in glucose and we convert it into cellular energy, ATP. And so go ahead and skip through all of this. This is just another example of a reaction that helps us breathe correctly, but for time we'll skip through all of that. And so this is when we led to this. In biology, there's uphill reactions and downhill reactions. Reactions that take a ton of energy because the energy is stored in the products, and reactions that happen relatively spontaneously, okay? And so chemical reactions that release energy often occur on their own, and chemical reactions that absorb energy will not occur without a source of energy, okay? And so this is the slide that we did a lot in class. And so activation energy, notice each one has like this little, bump to go over, but this one needs a ton of energy. Why? Because the energy is stored in the products. So notice how the energy is really high here compared to starting low. This one, the energy starts high, and so energy is released, and therefore this one is called exergonic. So exergonic, I like to think of energy exiting, and endergonic, which means energy has to be input in or goes in to the situation. Again, if you relate to the other terms, this is usually an anabolic reaction, and this is a catabolic reaction. And so every organism, of course, must have a source of energy to carry out chemical reactions, from plants using the sun to koalas eating the plants. Activation energy is this energy that's needed to start a reaction or get started. It's like a little bump, little push downhill. And so activation energy is important because you can see activation energy differs between absorbing reactions and releasing reactions. And so whether or not, whatever reaction we're talking about, and activation energy is still involved. And so that leads us into enzymes. Enzymes speed up chemical reactions because they lower activation energy. They make it easier for reactants to come together, and therefore they speed up chemical reactions. This speeding up, and it'll be on another slide, but this is called a catalyst. Okay, and so things can affect enzyme activity, and we'll look at this in lab in class, but temperature, the environment, specifically pH, even molecules that can block enzymes or interfere with how they work. Of course, human body cells, 37 degrees Celsius, they need to be able to work around that. And so here's an example of a picture of an enzyme. This is maltose, a sugar, and then this enzyme breaks it down into glucose products. So if we're breaking down, what type of reaction is that? And hopefully you said catabolic or exergonic, okay? And so in class, we talked about this example here of lactose-free milk. And so lactase, notice ASE, enzymes end in ASE. This is how enzymes are written in biology. And so lactase breaks down lactose into glucose and galactose. But if you don't have lactase, this increases, leading to fermentation, which causes a lot of side effects like bloating, gas, diarrhea, stuff like that for people who are lactose intolerant. And so these molecules, proteins pump these molecules into the blood, and then they're used for reactions, and especially cellular respiration. And so if you can drink milk without a problem, if you're lactase persistent, which means you're still making lactase as a young adult, you're actually a mutant because most of the world's population can't. And so we'll talk about this in evolution. But this lactase persistent gene arose in different places, specifically Europe and Africa. And so it's just kind of cool to think about, like, wow, like this, this all ties together with some huge themes. And so here's this term catalyst again, which means they speed up the reaction. How do they work? They lower the reaction's activation energy. So you can see right here, the red line is the one with the enzyme. And so you can see how it lowers the activation energy. And so this reaction right here, in which carbon dioxide combines with water to travel in the blood so we can breathe out this waste product, happens so slow that we wouldn't be able to maintain homeostasis. But this carbonic anhydrase, notice the ASE, speeds up the reaction by a factor of 10 million so that the reaction takes place immediately and carbon dioxide is removed from the blood. And so enzymes are very specific, which means they bind to only one substrate, or they only really do one chemical reaction. And so carbonic anhydrase gets its name because it also catalyzes the reverse reaction that removes water from carbonic acid. And so enzyme substrate. So how enzymes work is there's a site, and it's called the active site. And this is where the reactants, another word for reactants, I want you to know, is substrates. And so substrates can be brought together to react. So such a site reduces the energy needed. And so here's carbon dioxide and water. And so they would eventually combine, but it would take kinetic energy, it would take a lot more time. What the enzyme does, it provides like a little pocket or a little site to bring them together, thus making it easier for the reaction to take place, and therefore the products are released. Carbonic acid right there is the product. And notice that the enzyme is able to be reused. Okay, and so here's just some examples of enzymes. Amylase, breaks down starch, and here's maltase, sucrase, lactase, notice all the ASEs, breaking down disaccharides and polysaccharides. And so there's two analogies to how an enzyme works: lock and key and ball and glove. Basically, many keys can fit into one lock, but only one lock only one key turns that lock. Much like many substrates will bind or try to go into the active site, but only one fits. Only one is able to bind, and so that is what the enzyme catalyzes, that reaction it catalyzes. Also, a ball and glove is induced fit, which means an enzyme, the ball is a substrate, and so this is the enzyme, the glove. And so once the ball goes into the glove changes shape, and so it's a perfect fit. So that's called induced fit. And so here's a good picture here. Enzymes work by lowering activation energy. So notice here's the active site, here's the substrate, it binds, and then the enzyme changes shape, that's the induced fit. And then there's the enzyme substrate complex, and then you release the products. Again, this is breaking down, so this would be a catabolic reaction. Enzymes are affected by many different things. There's usually an optimum temperature for an enzyme, so you can see it's too cold or too hot, and that will lead to something called denatured, which means that this protein right here no longer works because it doesn't have the right shape. And so shape, remember, super important. Shape determines function. And so if you don't have the right shape, you don't have the right function. So here's the Antarctic ice fish, the one that has the clear blood. The enzyme acetylcholinesterase has an optimal temperature of -2 in the nervous system of the Antarctic ice fish, but an optimum temperature of 25 degrees Celsius in gray mullet found in the Mediterranean. So, of course, the enzyme probably has a different amino acid structure. It's very similar, but it's not the same because of the environment, right? Here's pepsin. Here's trypsin, urease. These are enzymes found in specific parts of your digestive system. This one's in the stomach. Makes sense, right? Because it likes a lot of hydrogen ions. It likes a lot of acid, okay? So that is lecture 2. Hope that was helpful. Please let me know if you have any questions. I hope you and your family are doing well. God bless.